Welcome to the debate. Right now, inside a single cell in your body, there are over 100,000 metabolites colliding, reacting, and, changing form every fraction of a second.
It's just an absolutely incomprehensible system.
It really is. And for decades, scientists have been trying to map this absolute chaos using, basically static, blurry snapshots.
Yeah, like trying to understand the traffic of a whole city by looking at a single photograph.
Exactly. But today, we are generating petabytes of multidimensional data per experiment. I mean, it's a staggering amount of information, and we are deploying artificial intelligence to actually make sense of it.
Right.
So we're drawing directly from recent analyses of global biological data initiatives, multiomics integration, and AI-driven precision medicine to ask a fundamental question about what this integration actually means.
Which is a huge question.
It is, and I hold the position that AI represents a true paradigm shift in biological comprehension. These tools, like graph neural networks and transformers, they aren't just sorting data. They are actually decoding the hidden language of the cell and revealing novel biological truths that were entirely invisible to us before.
Well, I come at it from a different way. I take the position that while this is obviously a monumental leap forward, AI's true revolutionary impact is strictly functional.
Functional in what sense?
In the sense that it is an unprecedented optimization and scaling engine. Yes, it accelerates existing pipelines, and it processes massive amounts of multidimensional data in ways human brains simply cannot. But it's doing this rather than offering us a fundamentally new biological philosophy.
I think that's heavily underplaying it.
I don't think so. We are drastically scaling our capabilities, sure, but we are not reinventing the nature of biological understanding itself.
Okay, well, to understand why I see this as a paradigm shift, we really have to look at the baseline of where we came from. Historically, traditional wet lab techniques, things like mass spectrometry or nuclear magnetic resonance spectroscopy, those were our gold standard.
Right, the classics.
The classics. But they only gave us those static snapshots we just talked about. You could see what molecules were present in the cell at a given microsecond, but you couldn't see the dynamic flow, you couldn't see the causality.
Which is the most important part.
Exactly. AI models have fundamentally changed this by treating biology like a language. Specifically, when we look at transformer models.
You're referring to the exact same architecture used in large language models, right?
Yep, the exact same architecture borrowed from natural language processing. So in a language model, the transformer predicts the next word based on the context of the whole sentence. It knows the word bark means something totally different next to tree than it does next to dog.
Right.
When applied to biology, these transformers decode sequential enzyme interactions akin to sentence structures. They predict the next metabolic reaction based on the cellular environment. By doing this, the AI uncovers what we call hidden stoichiometries.
Let's break that term down for a second, because hidden stoichiometries can sound a bit, abstract to someone outside the lab.
Fair enough. Think of stoichiometry as the fundamental math of chemistry, like how much of molecule A you need to react with molecule B. A hidden stoichiometry is a relationship between molecules that don't directly interact, but they mathematically influence each other through a massive invisible web of reactions.
Through downstream effects.
Right. For instance, these models are revealing precisely how tiny strands of genetic material, microRNAs, tweak enzyme kinetics far downstream in cancer cells. These are mathematical relationships that just completely transcend human cognitive bandwidth. We aren't just doing math faster, we are establishing an entirely new structural understanding of cellular biology.
I hear you, but the brilliance of artificial intelligence in this field lies in its scale and its capacity for dynamic adjustment, not in philosophical comprehension. Let's look at the actual foundation of this AI revolution. Where is the AI learning this so-called language?
From the omics data.
Exactly. It is entirely reliant on massive, aggregated data sets from global human initiatives. We are talking about the Human Cell Atlas, the Metabolomics Workbench, the European Human Metabolome Database. Without these monumental, entirely human efforts to standardize and aggregate this omics data, the AI has absolutely nothing to read.
But reading that data is exactly what unlocks the paradigm shift.
It unlocks an engineering triumph, absolutely. Consider AI-enhanced flux balance analysis, or FBA. Historically, FBA was how biologists tried to mathematically balance the budget of a cell, mapping what goes in and what comes out. Now, we use deep reinforcement learning to dynamically adjust those parameters in real time. That is a profound achievement in computational optimization. But optimizing constraints to predict metabolic yields is a functional output. It does not mean the algorithm has formulated a new biological definition of life. It just means it's found the most statistically probable path through a massive dataset.
Let's dive right into that concept of modeling cellular dynamics, because I think the structural representation of these models is where the paradigm shift is most evident. You mentioned optimization, but let's look at graph neural networks, or GNNs.
Sure.
In a GNN, we aren't just feeding an algorithm a flat spreadsheet of numbers. We are representing molecules as nodes and the chemical reactions as the edges connecting them. This inherently maps the topology, the actual physical and relational shape of the metabolic network.
Right, so it basically turns the cell into a massive interconnected web.
Exactly. It visualizes the cell as this bustling global city. You've got mitochondria as the power plants churning out ATP, the endoplasmic reticulum assembling lipids, and the results of mapping that web are just staggering.
The predictive accuracy is high, yes.
It's incredibly high. We are seeing predictions of flux alterations with 95% accuracy. Classical kinetic modeling could never dream of mapping the dynamic traffic of our biological city with that kind of precision. It isn't just balancing a budget, it is the first time we have a tool that actually mirrors the relational complexity of the cell in real time.
But look at how these models are actually applied in the real world to get those impressive numbers. You're talking about a city, but researchers at the Weizmann Institute in Israel developed AI-FBA to simulate bacterial metabolism under varying oxygen levels. Their model predicted ethanol production yields with error rates below 2%.
Which is unheard of.
It is incredible, but what is it mechanistically doing? It is pure parameter optimization. Or look at Ginkgo Bioworks in Boston. They are engineering microbes for insulin production. This is a massive boon for diabetes patients in places like India and Brazil, where the disease burdens over 100 million people.
And those yields are significantly higher because of the AI.
Yes, because the AI is optimizing industrial biological yields based on strict mathematical constraints. It is tasked with maximizing insulin output while minimizing toxic byproducts. Mechanistically, it is functioning exactly like a logistics algorithm optimizing delivery routes for FedEx or UPS.
Oh, come on, that's reductive.
It's really not. It is looking at the nodes and edges you mentioned and finding the fastest, cheapest route from point A to point B to increase a commercial yield. It isn't understanding the biological city. It's just bypassing traffic.
I see why you think that, but let me give you a different perspective. To optimize complex systems so flawlessly requires an inherent structural mapping that classical methods missed entirely. A logistics algorithm already has a map of the roads. In cellular biology, the AI is essentially building the map, discovering new roads, and identifying entirely new modes of transportation all at once.
It's still just finding the fastest route based on the data we fed it.
You cannot achieve a 2% error rate in a system of 100,000 interacting metabolites just by brute forcing a statistical trend line.
Why not, if the processing power is high enough?
Because biology isn't linear. The AI has to account for things like feedback loops and allosteric regulation.
Let's unpack allosteric regulation for the listener.
Right, so think of allosteric regulation like a remote control switch on an enzyme. A molecule binds to one part of the enzyme, and it completely changes how the other side of the enzyme works, which then cascades down the entire metabolic pathway.
Right.
When a graph neural network predicts how an entire pathway shifts because one single node is altered, it is demonstrating a deep comprehension of the system's architecture. The reason traditional methods failed to reach this level of precision is that human researchers couldn't define all those remote control switches. The AI is finding constraints and relationships we didn't even know existed. That is biological discovery. It is not just a logistical routing update.
I agree that it is finding the relationships, absolutely. But I maintain those relationships are statistical weights in a neural network, not a new biological philosophy. It's a highly sophisticated correlation engine.
A correlation engine that discovers new biology.
Let's test your theory then. If AI is truly comprehending biology rather than just optimizing a functioning logistics network, what happens when that network breaks down? Let's transition to precision oncology, specifically how AI handles the immense complexities of cancer and the Warburg effect.
I'm glad you brought that up, because the Warburg effect is actually a perfect example of why classical diagnostics fall short and why AI is a conceptual leap.
Go on.
For context, we've known for nearly a century that cancer cells have a bizarre metabolism. Even in oxygen-rich environments, where they should be burning energy efficiently, tumors choose to ferment glucose inefficiently.
Yeah, it's totally counterintuitive.
It is. It's like burning your furniture to heat the house when you have perfectly good firewood sitting outside. They do this to fuel rapid chaotic division.
Right, and it creates a highly chaotic metabolic environment.
Highly chaotic and highly variable. Conventional diagnostics completely miss the vast metabolic variability across different tumors. Enter AI. Look at the Deep Metabolism platform from Tsinghua University.
Okay.
It integrates multi-omics data from over 10,000 patient samples via the US-led Cancer Genome Atlas. By synthesizing that massive dataset, it classifies metabolic subtypes of cancer with 98% accuracy.
Which is great pattern matching.
It doesn't just categorize them, though. It identifies unique metabolic vulnerabilities, such as glutamine addiction and glioblastoma. The AI is literally discovering completely new biological targets for therapy because it can see the structural dependency a specific tumor has on a specific metabolic pathway.
That's a compelling argument, but have you considered that this is essentially advanced pattern recognition applied to existing therapies? The AI is incredibly good at matching a complex patient profile to a statistical outcome. But let's look at the mechanism. Look at the 2024 Nature Medicine study, a massive collaboration between the US, the UK, and South Korea. They used AI to predict patient responses to metformin,
which is traditionally a cheap, common diabetes drug.
Exactly. But the AI identified how to repurpose it as a metabolic disruptor in specific cancers, improving survival rates by 25% in clinical trials.
That's a huge breakthrough.
That is a massive clinical victory. But mechanistically, it is a data matching triumph. The AI looked at the tumor's metabolic profile, looked at the drug's mechanism, and matched them. Furthermore, look at how low-cost AI models are currently running on Google Cloud's AI healthcare platform. They are doing this to democratize precision oncology in sub-Saharan Africa and Southeast Asia.
Which is vital work.
It's incredible. The revolution here is about scaling clinical access. It is taking existing drugs, analyzing massive patient data sets, and matching the right compound to the right tumor at a global scale. It is optimizing the clinical pipeline, not necessarily discovering new fundamental biology.
But repurposing metformin as a metabolic disruptor in oncology wasn't obvious to human researchers, because the metabolic cascade that makes it effective in those specific tumors is simply too complex for classical observation.
And if you think metformin is just pattern matching, what about when the AI invents a molecule that has never existed in nature? If we move from classifying existing data to the true frontier, generative AI, the argument for fundamental discovery becomes undeniable. Let's look at neurodegenerative diseases.
A field desperately in need of acceleration given the staggering failure rates of classical drug discovery.
Exactly. The brain's metabolism is notoriously difficult to map. Take Alzheimer's disease. We know that plaques disrupt glucose uptake, essentially starving brain cells of their primary fuel. The Australian Brain Initiative has been fusing PET scans with metabolomic profiles. Through AI analysis, they revealed exactly how the tau protein interferes with ketone body utilization, meaning it blocks the brain's backup fuel source too.
Which is fascinating.
That alone is a massive mechanistic insight. But the defining moment, the real aha moment, is the 2025 landmark collaboration between Japan's Kyoto University and the U.S. firm In Silico Medicine.
Oh, right. This is the mitochondrial study.
Yes, they didn't just screen existing drugs like metformin. They used generative AI to design completely novel molecules targeting mitochondrial dysfunction. And these newly invented molecules were validated in organoids with 90% efficacy.
Well,
The AI didn't just match data. It understood the metabolic failure, and it synthesized a fundamentally new biological intervention to fix it. It acted as a synthetic biochemical engineer, creating a novel chemical reality based on its comprehension of the cell.
I'm sorry that I just don't buy that. Let me tell you why. The word comprehension implies a cognitive leap, when what is actually happening mechanistically is computational exhaustion. How did that generative AI design those novel molecules? By simulating billions of metabolic perturbations in silico. Billions.
It wasn't just random guessing, though.
It is computationally testing every conceivable atomic combination against a simulated binding site until the math works.
I think describing it as computational exhaustion ignores how generative models actually function.
Let me give you another example first. Look at Singapore's A-Star Labs. They are repurposing statins for Parkinson's disease via metabolic pathway optimization. They are using convolutional neural networks, or CNNs, to analyze microscopy images of metabolic hotspots.
Yeah, I've read about this.
By doing this, they are slashing development timelines from 10 years to under 2. Why? Because the CNN can process 1 million complex cellular images, while a human researcher processes 10. This is brute force computational processing, overcoming human cognitive limits through pure speed and scale. It is a triumph of engineering and processing power. It does not require a novel conceptual framework to hit a 90% efficacy rate. It just requires a fast enough processor to test every possibility until it hits the winning lottery ticket.
I think describing it as hitting a winning lottery ticket severely undersells the architecture of the models. Generative models do not just randomly generate billions of combinations like a brute force password cracker. They learn the underlying grammar of molecular biology.
Based on our data sets.
Right, but just as a language model learns the rules of syntax to write a coherent original paragraph, these AI models learn the rules of cellular stoichiometry and protein folding to generate a coherent original molecule. They don't test every combination. They predict the most viable pathways based on a learned structural understanding of the biological environment.
It's still a prediction based on weights.
When the AI at Kyoto University designed that molecule, it was applying a deep, internalized representation of how mitochondria function. That goes far beyond brute force. It is generative creation based on a sophisticated mapping of metabolic laws.
But the laws it is mapping are derived entirely from the human-curated omics data we feed it. Think about the National Institutes of Health funding projects at Stanford and MIT to ingest single-cell RNA sequencing data to map metabolic states. That is humans defining the biological parameters. The AI is finding the connections between those human-defined data points at a speed we cannot match.
And finding things we never could have found.
It is an extraordinary tool. But a tool that models an environment perfectly is still just a highly optimized map. It does not mean the map maker has formulated a new philosophy of geography. The outputs we are celebrating today, whether it's predicting ethanol yields with a 2% error rate, finding glutamine addiction in tumors, or rapidly identifying statins for Parkinson's, these are functional, practical, operational victories.
I think they're more than that.
They optimize the pipeline. They don't change the fundamental nature of what metabolism is. They just allow us to manipulate it at its native scale.
Well, I would argue that when a map becomes so detailed that it perfectly predicts and actively manipulates the territory, the map itself represents a new level of scientific truth. We are transitioning from a descriptive science of snapshots where we just categorize the blurry Polaroids of what we see into a predictive, structural science that actively translates the fundamental language of metabolism. The AI isn't just a faster lens, it's a translator for a language we couldn't previously parse.
And I maintain that its true triumph remains functional scale. By taking petabytes of multidimensional omics data, it dynamically optimizes diagnostics, yields, and therapeutic pipelines at unprecedented speeds. It takes the impossible math of 100,000 interacting metabolites and solves it so that we can cure diseases, engineer solutions, and distribute them globally faster than ever before in human history.
If there is a point of convergence here between our perspectives, it is the inescapable reality of our own cognitive limitations.
Honestly, the scale of this is where the human brain just hits a wall. Without AI, human cognitive bandwidth simply cannot process the petabytes of data generated per experiment.
Absolutely. And the sheer necessity of global collaboration to make this possible is undeniable. From Silicon Valley algorithms to European omics databases and Asian high-throughput imaging, this computational revolution requires a fully integrated global scientific community. None of these models work without the massive data aggregation happening across continents.
It forces us to reconsider the limitations of classical scientific methods. We can no longer rely on human intuition alone to find the patterns in cellular biology.
The data has simply outgrown us. There is much more to explore within this global genomic and metabolic revolution, and how we utilize these massive tools will define the next century of biotechnology.
At the start of this discussion, we talked about the cell as a system of incomprehensible complexity. For decades, we had nothing but a blurry, static satellite photo of its pathways. Today, we finally have a real-time, dynamic map of every interaction. Whether you believe this map merely helps us navigate the biological traffic faster, or whether you believe it fundamentally redefines our understanding of the system itself, is up to you. Thank you for joining us.